I was mortified. Anyway, but now I'm used to this. Anyway, another point is that 65 years ago, my advisor, William Lipscomb, that we heard mentioned, who actually passed away a month ago, for his thesis did the structure of methyl ammonium chloride. And so over sort of 4 or 5 decades, graduate students would go from solving something like the end of this lysine side chain to something like this. Of course, this was right after war. I guess nitrogen fixation somehow, at least in this talk, gets coupled to war. And so I'd always wondered what Lipscomb did during the war. And so it turns out that there was a civilian, national civilian chemical research effort that did a lot of work looking into things like various explosives, making a lot of poison gases to test gas masks and so on. And so Lipscomb was part of that project. So in his thesis, 2 parts are confidential, which now appeals to students. I think, oh, this would be great. I'd like to tell you, but so they also, at that time, we still have this tradition of having to do propositions that they defend before their thesis defense. And again, now they only have to do 3 at the end, which in my mind is 3 too many. But anyway, back then they had to do 10. 9 and 10 were confidential. But Lipscomb was an overachiever, so he has an 11th prop. Okay, 11A, research and study at the Institute have been unnecessarily hampered by the present policy of not heating the buildings on weekends. So this is sort of green energy. And the other, which maybe got me started on nitrogen fixation, manure should not be used as a fertilizer on grounds adjacent to the campus coffee shop. So anyway. Okay, so one of the really, I'd say, very satisfying parts about doing crystallography is it's model and hypothesis independent. And so we were interested in knowing the structure of the catalysts, and of course we were interested in that because no one knew what the structure of the catalysts were. And as was the case, and especially 20 years ago, the answer didn't just reveal itself all of a sudden, but it slowly unfolded, painfully slowly unfolded in a series of electron density maps at different resolutions. And so this was the first electron density map, and you can see you can't really see anything like atoms and so on at this point, but it actually gave us sort of an idea that there were 2 fragments that were connected together. When we actually were able to get the first structure, we had something that looked like this, which was pretty accurate in terms of topology, but the details in terms of bond distances and so on, especially in this region here, it turns out, I mean, so this is manual model building. The distance from here to here, those 2 atoms, is actually just about like one of these distances, but fitting these things by eye, we didn't do a good job. Then we finally got a higher resolution. You can see we were able to start making out individual atoms and so on. And after sort of 10 years, we were able to determine the structure at approaching 1 angstrom resolution, which at least technically, it wouldn't push this, but it's technically, we're getting at atomic resolution. We could start making out the detailed geometry. And so we have this movie over here. Essentially, we have, in this case, the irons are gray. Sulfurs are yellow. Sulfur's yellow. And so you can see there's like these alternating layers of iron, 3 sulfurs, 3 irons, 3 sulfurs, 3 irons, 3 sulfurs, and molybdenum. And the whole thing's really attached to the protein through 2 side chains, this histidine here and this cysteine up here. There's a citric acid derivative, or not derivative, it's a relative homolog, homocitrate, which also coordinates to the molybdenum, and it's absolutely required to fix nitrogen. If this is citrate, this won't reduce nitrogen to ammonia, it'll just reduce protons to hydrogen and so on. Well, one of the things that came out of this structure, besides getting more detailed geometrical parameters, is that we were able to establish the presence of this light atom, light meaning not a metal, in the center of this iron molybdenum cofactor, which is coordinated to these 6 irons in this trigonal prism arrangement. And all we can say at this point is it has density that's consistent with carbon, nitrogen, or oxygen. We have not been able to make more progress on this. And crystallography is actually not a very good technique, at least in proteins, for making this discrimination. I've always liked the idea that this was a nitrogen or a nitride that was left over from when it reduced the last dinitrogen that was reduced before we harvested the proteins and isolated the nitrogenase, but we don't know what this is, and that's one of the main areas of our interest now. Okay, there's also a P cluster. I mean, the nomenclature of this system is sort of odd. P stands for protein. The cofactor, iron molybdenum cofactor that we just looked at, is also known as the M center because it's magnetic. So anyway, but it doesn't get sort of the attention that the other metal cluster has, but this also has an odd structure that we've seen in different conformations that we believe correspond to different oxidation states. Okay, so I wish I could tell you how this enzyme works. This is my favorite example of just because you know the structure of something doesn't mean you know how it works. But it's also true that if you don't know what it looks like, you'll never understand the structure. And so just to sort of say a little bit about mechanism, there is almost nothing that's actually known. A lot of interest is trying to understand where substrates such as nitrogen or protons or acetylene bind. And there are two schools, and these are pretty passionate schools, even though there's probably because there is no experimental evidence. Everyone can feel right. So there's the molybdenum school and the iron school. And I should say I belong to the iron school, but I at least acknowledge that there is this other school that includes people like Dick Schrock, who won a Nobel Prize a few years ago. Okay, so anyway, this sort of basic questions as to how nitrogen binds, whether it's to one iron, multiple irons, the role of these residues. There's a lot of protons that have to be transferred, and probably things like this histidine and maybe the homocitrate play an important role in the proton transfer process. There actually are pretty interesting parallels between how the central ligand interacts with, say, these three irons or these three irons, and the Haber-Bosch catalyst that is an iron catalyst that really hasn't changed much in the 100 years since the Bosch really did an exhaustive search. And so I think that may not be a coincidence. There may be some mechanistic parallels between the industrial process and the biological process, but this is still all speculation. So, okay, yeah, there are experimental, mainly through the efforts of Lance Seefeld at Utah State, Dennis Dean at Virginia Tech, and Brian Hoffman at Northwestern, trying to use various spectroscopic methods to try to understand how substrates are interacting with the cofactor. Okay, well, the other protein is the iron protein, which looks like a large category of nucleotide binding proteins involved in signal transduction and other processes. One of the things that's fascinating, I'd say, about biology or structural biology is you can see how the same basic motif for architecture is used in a number of different contexts. In this case, the binding of nucleotide, ATP, to regions such as this P-loop and switch-2 region that are found in these large category of other nucleotide binding proteins, instead of being used in some sort of signal transduction pathway, instead it changed the environment of the iron-sulfur cluster that's involved in getting electrons from the iron protein to the MoFi protein. Okay, just one kinetic scheme. The importance of the iron protein is that it is the only known reductant that results in catalytic activity of the MoFi protein to reduce substrates to products. So most inorganic chemists, when you show them the system, they say all you need is a strong reductant in the MoFi protein and you can make ammonia, and it's not true. The iron protein, there's something about the way in which the timing and the way in which it transfers electrons to the MoFi protein that if you want to reduce nitrogen, you have to have both components. So the iron protein binds to the MoFi protein to form this complex, and it's in this complex that ATP hydrolysis is coupled to electron transfer. And so we've been really interested in understanding this coupling between ATP hydrolysis and electron transfer. So we were interested in trying to understand not only the structure of the two proteins, but also their complex. And so when I was a postdoc at the University of Minnesota with Jim Howard, amazing nitrogenase biochemist. And Jim was able to show that aluminum fluoride stabilizes a complex of these two proteins. And say it has been shown in a number of signal transduction proteins that aluminum fluoride is an activator. It basically looks as a mimic of ATP hydrolysis, and so it sort of gets stuck in the on state. I should also point out for the chemists, real chemists, get very unhappy when they see aluminum fluoride because they say that'll look like methane, and this is aluminum fluoro, I don't know what it's the proper terminology, but we use it sort of cavalierly, and so any real chemists, I apologize for misstating this active species. Okay, so if we now take half the complex and sort of cut through there, you can see that just from the metal centers, there's a clear evidence for an electron transfer pathway going from the iron-sulfur cluster through the P cluster into the FeMo iron molybdenum cofactor. And one of the parts that we've been interested in, that I'll say a little bit, is how ATP hydrolysis at this point, can end up triggering the transfer of electron from here through the interface between the two proteins and end up at the iron molybdenum cofactor where it can reduce substrate. So there's these, all these sort of long range effects where conformational changes can get propagated over longer distances. Okay, the complex between the two proteins, as I mentioned in the purification, these are very negatively charged proteins, and that's indicated in this color coding by red. So red is negative, blue is positive. It's not Democrats and Republicans or any of this stuff. Okay, so these are very acidic or negatively charged proteins, but they're not uniformly negative. Instead, there's localized nature of the patches, and the way this is represented is taking this view here, and this is the iron protein, this is the MoFe protein, then sort of turning them sideways and opening it up and looking at the interface. And so this is where the 4 iron cluster of the iron protein is. And so if you can imagine, you take these two surfaces and just put them together like closing a book. And this region here, which has some positively charged groups and is neutral, ends up overlapping with the negatively charged regions of this interface. And the negatively charged regions here end up interacting with the positively charged parts here. So there's strong electrostatic stabilization of these, of this complex. I mean, you can't really tell from this, and probably a computational chemist would have enjoyed sort of thinking about this in more detail. But you can imagine that if these don't fit exactly, that there might be some tendency for them to tilt back and forth, where maybe on one side this part could interact, and then it could rock over to the other side and interact with these. And so actually, Akif Tezcan, who's now at UCSD, has been able to do a number of structures in different nucleotide states of these proteins. And we do see this sort of rocking motion. And I think that there's not one sort of favorable electrostatic interaction that stabilizes one form of the complex, but instead there's actually changes in the iron protein conformation associated with this. They're involved in electron transfer and ATP hydrolysis. So if we look at all these complexes, so for electron